We can start a recording. Thank you. And hi, everyone. Welcome to another Applying Ethology webinar series hosted by the Animal Welfare Slack workspace. My name is Jianyan Chu, and along with our managing team members, Christian Nawers, Laura Welling, and Sebastian Gumon, we really thank you for joining us on your busy Monday. And we're really happy to see many of you tuning in today. So before we start, it'd be great if everyone can double check your microphone is on mute and your camera is turned off. And please, at the end of the talk, type your questions in the chat box so we can go through them at the end. And so today, I'm very excited that our speaker is Dr. Océane Schmitt. And Océane, we have known each other for so long, so I'm really happy to invite her to talk today. So she's a pig behavior and welfare researcher, and she did a PhD at Chagas in Ireland. That's where we first met. That was already 8 years ago. And she had many international experiences working in New Zealand, Canada, the Netherlands, and the UK. For example, she worked at Bristol University on rodent behavior and welfare. And currently she's working at the MedMed Uni Vienna in Austria. Because of her mixed background in agricultural sciences and ethology, she really likes to use applied and fundamental research in her projects to promote animal welfare. Her PhD was about improving the survival of piglets during lactation, but she also investigated the cognitive abilities of pigs, their emotional states, and more recently, how positive human-animal relationship affects pig behavior and physiology. And she's also working with the French Association Sphere to encourage the rehoming of laboratory animals and improve the ethics of research. So I will stop here and I will hand the floor to Océane, and really looking forward to your talk, Océane.
Thank you, Jen. Great, perfect introduction, I would say. So I wanted, so Jen did it really well, but if you want the names of or like a bit of my CV, I wanted to first start with where I come from. So in terms of countries as well as background, like scientific background. So I started as an agronomist in France, and that's where I was first taught about animal welfare with the farm animal welfare. And I got totally passionate by the topic. So I decided to specialize more in it. And I went to the Netherlands to do another master degree. And I did two internships, so one at Wageningen and the other one in Canada. Both in research, and I loved it so much that I decided I wanted to do a PhD. And that's where I went to Ireland. After that, I did two postdocs, and one took me away from the pigs. So I started with cows in Wageningen, then worked with pigs in Canada, and in my PhD, then mice, and I'm back with the pigs now. So I went through across different countries, species, and types of fields, I would say. And that's what brought me to wanting to save piglets in the first place. So in my PhD. So my presentation is not going to be chronological, it's going to be more with like by topic. So don't get lost in there. During my PhD, I investigated management strategies to help piglets born in large litters to survive or to optimize their survival. And that's because nowadays the selection for large litters has made the sow giving birth to more piglets than they can actually raise. And that's of course an economical concern, but a welfare concern for the sows, the piglets, and it's also not looking great for the industry that babies are dying. So we investigated three types of strategies. So the nurse sows, which consist in taking a sow that has already reared her own piglets, and she will adopt newborn piglets that are extra in their own litters and rear them for another lactation. We investigated artificial rearing, which consists in taking a whole litter away from the mother and rearing them with ad libitum replacer in a dedicated enclosure. And finally, we wanted to give an energy boost to the little ones, the smallest and weakest piglets in the litter, so typically under 1.1 kilo, and see if that could improve their survival. And I'm going to describe for each of them like more the story behind. So you won't see all the results, but the papers are there. So it's I'm just one Google Scholar search away for your if you get curious about everything. So the nurse cells, the way they work is that you have this cell 21 days into lactation, and you wean her piglets, and then you give the biggest, like typically the biggest ones that are born in large litters, and you just take all the rest, like in several litters that are born on the same day, and you complete up to 10 or 12 piglets. And in the two-step strategy, as transparent as it is, you have two nurse cells. So the first one, again, is 21 days into lactation, and she has a piglet twin. She gets to adopt 7 days old piglet from the second nurse cell, who can then get the biggest piglets from the large litters that are born that day. And in my trial, I also had two types of control, which represent the reality of farming. So the first one was the sow will remain with all the piglets will remain with her, so she only gets her own biological piglets. And in other litters, the piglets were cross-fostered, meaning that some of them were exchanged so that the weights will match better. And just to give you an overview of all what we investigated, so we took behavioral as well as physiological and let's say more physical measures, and of course, like mortality was one of our focus. So I didn't find differences in mortality, very little in weight, and body lesions of the cells. And the behavior around fostering was pretty much the same. Like everyone investigated each other because most of them were new to the cell. But what I saw was higher, like a more frequent or more frequent fights in the cells, in the litters that were fostered onto the cells that were in late lactation, compared to the ones that were in early lactation, so the ones that were just borrowed and the ones 7 days into lactation. And that might represent that the piglets were at the sow stage need to match, like the piglet's age and the sow lactation stage needs to match. Also during fostering, I saw a difference between the sows that were 7 days into lactation when they were when their piglets were taken away and she was giving new ones. She had the highest increase in cortisol, so that could represent more stress compared to the others, and that would make sense because she was just after 1 week raising them, so the bond is stronger because they are more reliant on her for their survival. With artificial rearing, so it's often done as acting as a second nurse cell. So you take the 7 days piglets from the cell that will receive the newborn, and you put them in a pen where they have light, heat light, and a libitum milk replacer. And for this study, I looked, of course, at mortality and weight, but also I looked at behaviors such as play, aggression, or aggressive behaviors and abnormal behaviors such as belly rubbing, because those I was very curious if the behavioral development of those pigs was the same, as they don't have access to a mother and they don't grow in the same type of environment, which is typically smaller in the artificially reared. I looked at their emotional state and the lesion and the health. I found differences pretty much everywhere in all my measures, but the ones that I see more telling the story are these ones. So I saw more ear and tail biting, so more, let's say, manipulative / aggressive type of behaviors. And also a lot of belly nosing, which is supposed to represent the needs, an unsatisfied need for massaging the udder and performing a normal nursing, which is normal because they couldn't. And that was reflected in the weights. So gradually, my pigs also, like both started in both groups, they started with the same weight, but gradually the piglets in artificially reared didn't lose weight, but they didn't grow as well as the salary one, and that ends up in a big gap at weaning, which is important economically for the farmers, but also for welfare. It reflects that they were not feeding well enough, which is surprising because they had a libitum milk replacer, so that was against all expectations. And that was maybe because they were nursing-related displacement. So it means that the piglets were pushing each other at the milk replacer truck, and that more than they would fight at the sow udder, and that would reflect a problem with feeding, that it wouldn't allow them to feed the way they want. So after that, we looked at the efficiency of giving a fat-based energy boost at birth to piglets that were 1.1 kilo or below at birth. So 3 hours postpartum, I gave them 2 milliliters of either water, so with a zero, like with no energy content, but would fill their stomach, coconut oil, and a commercial energetic product made for piglets that had the same value energy. And there was a forced treatment where I just handled the piglets, so I pretended I was dosing them, but I didn't give anything, and that was to see the effect of stomach feel with zero energy. And I looked again mortality and weight. We also like the rectal temperature change across the hour post supplementation, and also the blood glucose concentration change. And I didn't find any differences between the different treatments, except the blood glucose gain, which tells quite a nice story and such. So the coconut oil was the highest compared to the others. But what's interesting for me is that the water treatment, which basically was filling the stomach of the piglets without giving them any energy, those ones, they actually lost glucose. So that means that I could trust more what I was doing, in the sense that it went in this prediction that if you fill the stomach, like the 2 milliliter was enough to fill the stomach somehow. And if it had no energy value, it will actually be not good for them. So the conclusion of my PhD was that basically nurse sows, well, they do work, but it doesn't seem great either for the pigs or the sows, but it doesn't seem so bad either. Artificially rearing does not work, and I would definitely never recommend it to anyone to do it. And supplementing with piglets at birth doesn't work at all. It doesn't work much. I mean, the blood glucose gain was not reflected in higher weight or higher survival, so that's not efficient. So then I was wondering, what should we do? And while writing my thesis, I was asking myself the question: is it ethical to harm one individual to promote the survival of others? And that's a very open question still nowadays. I don't have one. I'm still wondering. But I thought I would share it with you in case someone has a bit of answer for me. And then I thought, is there like one great solution? And I actually think there is. So there was another PhD student at the time that investigated the use of milk cups in souring pans. So it's basically taking the cups from the artificial brewing system and giving them into the normal souring pan. So it allows to keep the whole litter with the sow. So the sow doesn't have to go for an extra lactation. The litter stays with their mother and the siblings, and they have a chance to have access to milk if they miss a nursing bout because there are too many at the udder. And I think that's a system that should work well, and that should be recommended. So during my PhD, I also had the pleasure to play with a quite new technology called infrared thermography. And with this special camera that takes a fancy colored picture that reflects the temperature of the animal, the emitted temperature of an object. I investigated the thermoregulation at birth of the piglets. I paid attention especially to their ear base and ear tips, because the ear base is close to the skull and it should reflect internal temperature better, and the ear tips are extremities that the animals and piglets in particular will tend to redirect blood flow to redirect it to the inner organs and increase the core temperature. So they would reflect thermoregulation dynamics. And I did two studies, one at INRAE comparing two genetic lines selected for feed efficiency, and one at Chagas comparing piglets with different level of intrauterine growth retardation, IUGR. And IUGR is the piglet on the left. It has a dolphin-shaped skull compared to the piglet on the right, who has like more straight line between the skull and the snout, and it has also like the IUGR piglets on the right also has more bulging eyes, so more big bulging eyes. That's how you recognize them. So the feed efficiency study was pretty straightforward. That the piglets, you can see the ear tip on the right graph, and you can see there is a decrease in temperature at 15 minutes postpartum in the low food efficiency piglets. And that means that those piglets experience a bit of a hard time thermoregulating, and the others, the high food efficiency, they just kept going straight up, which is what you would hope your piglets are doing. So that was, yeah, nice results. And then with the intrauterine growth retardation, I also measured, so with the rectal temperature compared to the ear base, and I saw the same path and the same order of pigs. And you can see that the highest score, which is score 3, the red line below is less efficient at thermoregulating and doesn't reach the temperature that around 37, 38 degrees that the piglet should reach by the 1 hour postpartum. And because I love cognition and I had time, or I made time, I decided to take those piglets, the small piglets from the experimentation study, to test them in cognition tests. And I used spontaneous object recognition tests, which consist of presenting two similar objects once to the pig, then you take the pig away for 45 minutes, and you bring it back to the pen, and one of the objects has been replaced. And if the pig remembers that he's seen the first object, then he would investigate the new one more, so either first or more, because pigs are neophilic and they would be attracted to novelty. And then you compare the time spent with each object. And the double T-maze test, so it's basically a cross from two extremities, so I will show you a video. So the pig can start from the south or from the north, and that's for them to learn not turn always right or always left, but pay attention to the white stripe, if you can see on the west arm. The west arm is the rewarded arm for that pig, as you can see. And so when he makes the good choice, he gets to have access to the reward. The other arm is also a reward, but it's inaccessible. So at some point, it has to pay attention like this, and maybe remember, like, understand that he has to pay attention to the white stripe to turn right. So this pig is not very successful. I mean, he's in the stage of learning. It's just one nice video that you could take. But as you can see, it's he doesn't really pay attention much to the stripe. But here is the moment where he does, and he pauses and he wonders, and then he makes the good decision. And that's for those moments that I love cognition. I love seeing animals thinking and then understanding what's happening and what they should do. But yeah, as you can see, it's not always great, and sometimes they pay attention and they seem to understand, but then they fail again. So yeah, that's the reason why I love cognition, to try to understand all of these processes. So with this T-maze test, what I saw is that the piglets with the score 3 of IUGR, so the most retarded, intrauterine retarded, growth retarded, sorry, they actually were better, like shorter to enter the successful arm, meaning they made faster good decisions. Even though the number of sessions to reach each step was the same, they seem to process information maybe better than the two middle scores, at least, and more or less as well as the non-retarded ones. And that I also saw in the spontaneous object recognition test. So what the graph shows is that the blue square should be round zero, because the two objects, or it can be different if there was a flat side preferred by the pig. But basically, you should see a difference between the square and the blue square and the red lozenge. And that's what you see with the non-so retarded piglets, and that's what you see with the heavily retarded piglets. And that's what's really interesting, because it seems that there is a protective something that happens, but during the growth retardation process, they also preserve their brain development somehow. And that's actually something that exists in humans. So my idea was that probably that's what happened with the pigs, and I found it amazing. And then the second part of my career maybe is cuddling pigs. And then I started with my master's, so back before the PhD, of course. And that's so when I went to Canada, and I worked on a study that was looking at the effect of quality of human-animal relationship. And for that, we had a nice treatment. So you can see the young me petting pigs and enjoying it. So 2 minutes a day for 5 days a week, pretty nice for them and for me as well, besides being beaten, as you can see. And there was a negative treatment that was supposed to reflect what could happen on a farm. So like, running or catching pigs, maintaining them, like restraining them for a bit, playing, well, not playing, but like banging the walls with this paddle, which is used usually to move them around. So that was negative, but not aversive in the sense of doing harm to them. And then we trained them in a go-no-go task. For the judgment bias task. So judgment bias task, I guess you have heard of it, but you teach one cue to the pig. And then you can decide to approach or not. So that one was a positive cue, meaning there is food. You could see it was a shy approach, right? The butt of the pig stayed outside. It stretched. And then you teach the opposite. The opposite cue will predict an opposite treatment. And once the pig have learned that, then you test them with median cues, and the most median is like really in the middle of the two that we taught them. And then it's ambiguous, meaning they have to make the choice. Do they think there's going to be a reward? In that case, they're optimistic. Or is there going to be a punishment and they don't approach? And in that case, it means they're quite pessimistic about what's happening. And what we saw was great, was exactly according to predictions. So that the pigs that were in the gentle treatment, they were more optimistic than the piglets that were in the rough treatment. And the minimum treatment was just minimum contact, so basically just routine treatment, but no interactions with the pigs. But what was more interesting than that is that when we included a human observer in the test, the negative treatment pigs, the rough handling, were actually more pessimistic than without the human observer, and they were more pessimistic than the neutral pigs, which were the most pessimistic without the human observer. And that was very interesting to see the influence of a human observer on the result of the test, depending on the relationship they have. And probably that's what led me to my current position, where I investigate positive interactions with pigs. So now we only cuddle them. There is no rough treatment anymore. And we want to characterize the positive interactions from the point of view of the animal. So when something is positive, what's going on, basically. And for that, we look at their behavior. So of course, the time in contact, but also the engagement in the context, seen as the reciprocity and the synchrony of the interactions, but also some vocalizations and general behavior. And we look at the physiology as well, so heart rate variability, the secretion of neurotransmitters, and the brain development and connectivity, and their immune competence. So we are currently looking at the data, and I hope to come back very soon and give you the results, or maybe my colleague will give you the results. Let's see. So first, we investigated the standardized versus free-form treatment interaction. So as you can see, I will play both together. So in the free-form, basically everything can happen, but my colleague stays in one spot and the pig approaches as she wants and interacts in the way that she wants. In the standardized one, my colleague has a schedule, so she gets up, walks to the pig no matter where the pig is, and then impose a series of strokes in 1 second per stroke in every second. So it's very standardized, very, let's say, robotic. And we want to see if that changes the synchrony and the reciprocity of the interactions and their general behavior. Then we looked, and we are currently looking into the dynamics of neurotransmitters. So in the first study that we did, we compared, we tested pigs in free-form or ignoring situations. So ignoring was just to have a neutral, like a neutral presence of the human, but really nothing was happening with the interaction. And can see the videos are lagging a bit. I'm sorry. And in the in the positive interactions, it was a free-form contact, and you can see. So the blue overall is the unfamiliar human, and you can recognize my my colleague in the black overall. And what we wanted to do to see in this, and then you can see the pigs have a pouch, like a little square on their butt, and that's to hide their catheter. They have a spinal catheter that allowed us to measure oxytocin, dopamine, serotonin, and opioids in their as they're interacting and after they interacted. So that's measuring what's happening in their brain, basically. What are they signaling? And they have this little belt around the girth that is measuring the heart rate. And in the trial that we're currently having, we are looking at blocking the receptors of dopamine and opioid and see how it affects their behavior in the free-form type of contacts. So we think that blocking the dopamine will remove the motivation to interact with the human, and we think that the opioid blocking the opioid receptor will block the pleasure that they have in the interaction, and therefore they will still interact, but maybe not engage so much. But let's see. I looked at the brain development and immune competence of pigs also during long-term interactions. So for 9 weeks, sorry, I went 3 times a day on the farm, and I interacted with a group of pigs, 2 sessions per day and 10 minutes per session. And at 5 weeks, so before the treatment and after the treatment, we put them in an MRI. I'm not lying, it's a pig in an MRI, and we scanned their brains to see what was going on, and especially when I was stroking them on the back to see the brain response to being stroked. And then I submitted them to the LPF challenge, which consists in giving lipopolysaccharides, so it's the shell of a bacteria without the. So the pigs will get sick because it will signal to the immune system there is a bacteria inside us, and we need to get rid of it. But they would not develop a disease because the bacteria cannot reproduce and contaminate on the long term. So it's, let's say, pretty safe for the pig, but unfortunately it makes them very sick. So I looked at the sickness behavior across 6 hours that the pigs are sick for, and then after they recover. And I took blood samples to look at markers of the response of the immune system. That leads me to the last part of this talk, which is what I've gone through since I started science, is being ethical. And all the questions about ethics in research. You could have, you guessed that already from the conclusion that I gave of my PhD. But that's also why I'm interested in mixing fundamental and applied research. I do think it's a way of optimizing the way we have. We produce results, but also it's using the animals as much as you can, and it's I don't know. For me, it's a way of doing better science, but that's my own opinion. And one example that for me that leads me into this meeting is that these have excellent cognitive abilities, and we can use them to improve their welfare if we understand them correctly. We can improve the way they live if we understand the way they think. And that's true for all species. I'm just taking the pigs as an example because I'm more of an expert in them, but I definitely prone this for all species. And conducting ethical research has been a growing concern, let's say, for me across the years, across the trials that I was doing, probably because I was I was coming from observing situations to making situations, and I think that made me question the way I was doing things. So as I say, like maximizing the data collected in one trial is really important for me. And thinking of the 3 R's, when we think of our experiments, we have to, like, I think, what are we imposing to the animals? What are the actual costs, and will the results be meaningful for the animals or for the humans? And because I recently, since the postdoc I did in Bristol and where I worked with mice, I was confronted with the fact that at the end of an experiment, if the animals cannot return to what is their normal life, what we call normal life as a livestock is farm life, so production. But that's a convenient end for people working with livestock species, because in the end, it's they come back to the purpose for which they were bred, or they were born. But with mice, for instance, you have to buy them, you have to order them, and that made me question if we should actually kill them. So we rehomed them, and that was great. And that's how I got started with the idea that we could rehome, or we could consider rehoming animals if they are not, if death is not required. Because the end of life is a more burden for the researchers and also for the people who have, so the researchers who have to make the decision and the people who have to carry out the procedure, which can be the researchers, but also the technicians or the vets, which none of those people have chosen their work to kill animals. So we have to understand the psychological impact that it can have. And therefore, rehoming can be the 4th R in ethical research, although it cannot be, I don't think it can be imposed as such, because the reality is that there are limited adoption opportunities. For instance, some universities or researchers fear to be exposed to activists and violent activists. Some people are still traumatized by violence that was done to researchers in the UK in the 60s. And a lack of information to the public about rehoming and adoption doesn't make the public very inclined to adopt animals that have already been used. And I think the non-transparency of research doesn't help the situation, because people imagine that we do research. You can ask ethologists that sometimes we just sit on a chair and watch animals for hours and we don't do anything to them. And they still see those animals as having endured a life, a bad life, or having trauma, and therefore it can be hard to convince people to also adopt those animals. And finally, there are a limited number of suitable places outside of personal adoptions, like shelters and rehoming centers are pretty rare. And we have tried so hard to rehome our pigs, but it's virtually impossible, because pigs live a long life, and once a mini farm has one pig, they're good for 20 years. So guess about rehoming 20 pigs that are just 5 or a few months old. Yeah, and there is a high number of animals, especially in the rodents. Although rodents can be good pets, so yeah, they have more diverse issues. Opportunities. So I'm just advertising quickly all the places that I've worked at. They are great places. I definitely advise them to everyone. If you are considering an internship, PhD, postdoc, whatever, I'm sure you can contact them directly, and those great people will answer to you. And now I would like to thank you for your attention and invite you to ask me any question that you have. And I also want to thank all the animals and the humans who helped me in my way. I didn't tell you about the cows and the mice today, but they're here at least for the gratitude. Thank you.
Thank you very much, Océane. And so please feel free to type in your questions in the chat box. So while we're waiting for a question to come in, I'm just the first question I have is you talk about like so many different studies, and I'm just wondering, can you talk a little bit about the last study you mentioned about the human-animal relationship study, and what is really the implications behind you, because you're investigating about the resilience, disease, and then all this physiological response. So what would be the implication for that?
So, I mean, the very direct implication would be that we could inform better the farm staff about what they should do. So for instance, I mean, it's pretty unrealistic to ask a farmer to go and sit with a group of 3 pigs for 10 minutes, twice a day, 3 times a week, because of the number of animals they have. But if we find that it improves their immune competence, for instance, then we have a starting point to tell them that maybe some contact would be better than none. And understanding if pigs like to interact with be free of their own choice, or if they like better to be struck in a certain and predictable way, then that's also something to inform to improve the way we interact with those animals. It's not only the farmers, it's also the researchers that are working in the field, because the thing is, and that's why this project was intended, is because we don't really know if the contacts that we give to the animals are positive. We assume they are because they are gentle for us, they are strokes, but we don't know how the animals feel. We know they stay, maybe sometimes they ask for more, but maybe sometimes not. And yeah, that's the aim of the project.
Thanks. And Lindsay has a question, and she said that could you explain more about the INHI results you show again, the HRFI versus LRFI? Maybe, yeah, also if you don't mind opening the slides again. Sure. I think she's just wondering about the hypothesized explanation for your findings.
Oh, right, yes. Oops, sorry. Here we are. Yes. Yes. Sorry. So I would first explain the letters. So HRFI is high residual feed intake. So it means that the pigs, when they eat, they have more. They gain less weight compared to the food they eat. So it's the food conversion ratio. So the ones that have a high residual feed intake, they are less efficient than the ones with low residual food intake. And so the idea is that, so the justification is that the ones that can convert better the food into muscle or into growing, basically they can do it from birth. And it means that when they get the colostrum, they can convert it already into energy and use it efficiently. And that's what we saw. And that's what I said, like the red line, which is the low residual feed intake, so high efficiency pig. They keep increasing their temperature all the time, which shows that it's a stable thermoregulation. It's the way it should be, that they increase and then they reach the temperature that they're supposed to have. But the less efficient ones, the fact that they have a gap at 15 minutes, so a decrease at 15 minutes and then they increase again, it shows that they experienced a moment where they needed to get all the heat to their core, meaning they didn't have enough heat to maintain it in all the body, because that's the ear tip. That's the place where the blood is the least, or the heat is the least needed. So if it decreases, it means that the inside temperature is maintained, but at the cost of the extremities. So it shows a less efficient thermoregulation. Was that clear? I hope that was.
In a sense, can we also say that the infrared thermography data can actually become a kind of a surrogate indicator that whether the pigs are growing like efficiently or not? In a sense?
No, not in that sense. No, so growing efficiently, definitely not. That's a very long stretch. Let's say it's because I knew what the pigs were meant to be that the data made sense. Otherwise, I would have told you I have two groups of pigs. One is more efficient at thermoregulating, the other one is less efficient at thermoregulating. But I cannot know if it comes from the fact that they're more able to convert food into energy, or if it's because they're better at keeping warm because they're smarter and they keep huddling instead of spreading in the pen and not paying attention to the warm areas, or like many other possibilities could be.
Okay.
Thermography is solely a measure of the temperature emitted by the animal. So unless you know everything about the context, it's hard to interpret other than a temperature emitted.
Okay, thanks, I understand. So Sarah has a question. Hi, Océane, great talk. She's curious about your results on artificially reared piglets. Was there more competition around the milk feeder compared to when reared with the dam that could explain your findings?
Absolutely, yeah, it's exactly what happened. So the thing is, the pigs, they love they love. I mean, that's the way they're made. What happened naturally is the sow feels like she needs to have a milk let down. So she will grunt, and that will attract the piglets. The piglets will gather the udder all together, they will massage the udder all together, and then the milk is ejected for a couple of seconds, and then that's it. So the piglets are genetically programmed to go as fast as possible to the udder, secure their teeth, because if they miss those few seconds of milk ejection, it's gone. I mean, it's over for the next hour. So they keep this willingness of feeding together, even in the artificially reared system. And what happens is that when what exactly happened is once we made the warm milk and we put it through the pipes and it was delivered, then the pigs wanted it because it's warm, so it's yummy, it smells good, it's more appealing. And of course, not like once one starts, everyone wants it. But those caps are really small, and they can have like 3-4 pigs maximum around them. So when you have two, basically only 8 pigs maximum could be around it. And you have 12. So 4 of them will just be pushing the others around, and it's just a circle of pigs pushing each other and trying to access these things. That's not something I anticipated, to be honest. But then when I saw it, it made total sense. So yeah, it explains my finding very well.
So following us,
And outside of those moments where the pigs were triggered highly to go and feed, they would not engage so much. But then once one will go and start drinking from the cups, most likely the 12, like the 11 others will get up and go for it as well. Sorry, what did you say?
Sorry, I was just, because you were saying about the number of cups, so they don't have exactly the same number of cups for exactly the same number of piglets.
No, they have only, I mean, the systems that I have seen have only 2 cups. And then you're supposed to put 10 or 12 or more piglets, depending on the age. But let's say 12 is the routine one. So it's not enough. It's enough if you think like a human. If you think, oh, it's ad libitum, they can go whenever they want. There is unlimited access to an unlimited quantity, so they cannot starve. They will feel great. But when you think like a pig, you go all 12 together to the cups and you fight for it. So it's not efficient at feeding, and they would like splash the milk and everything. So it's quite, it was a bit ridiculous, let's say.
So do you think that if we actually do have enough number, let's say 1:1 ratio pig and cub, do you think that the artificially reared ones will be performing the same as a dam one, or maybe they're more than just, you know, availability of milk? Maybe the interaction with the mother or some other thing also.
Yeah, I think there was way more at play than just this competition, because as I said, the milk was always there, so they didn't starve really. I think there was also other things, like for instance, they got sick a lot because the milk is very rich. It's much richer. Actually, it's not smell milk, it's cow milk. So it's not really adapted. It's not 100% adapted to pigs. And it contains pig plasma, that's to provide them with immune materials that they need and they usually get from the colostrum and the milk of the sow. But the problem, what I think, because it's quite a personal opinion and I think based on a couple of papers only, that it's too, yeah, it's too rich for the piglets, and then they have diarrhea because they just have, yeah, too a non-adapted feed basically. So they get more sick, and I think they lose weight also because of the sickness, and the environment is not perfect either. Like it's way too warm, I would say. Yeah, there are other, like, way more reasons why this system is not one of my favorites.
Thanks. So I know you need to leave on time today. I know we're still waiting for
we have time still.
the oldest historian more questions. If I may, just throwing a more difficult question. I mean, I guess we're all thinking about it as an animal researcher all the time, but because you're now, you know, promoting rehoming of our research animals, but of course at the same time we're still sometimes in our studies where we have to do some more invasive procedures in order to get the data we wanted. So I think for yourself as animal researchers, how do you kind of reconcile with this? You know, sometimes you have conflicts, you have to think about so many difficult ethical questions. So how do you deal with that? And then what advice you would give to, let's say, early career researchers?
Well, my personal thing is I had to live it to know it. So I think experiencing things is the best way to know where you stand and what you can or cannot handle. That's what life is for, right? I mean, it's experimenting. But I would say it depends on your philosophical or ethical point of view or stance, let's say. If you have a utilitarian point of view, like I do, you can reconciliate these things by thinking that the benefits outweigh the cost. and that during the time that the animals are under your care, you give them the best life possible. You do the best you can to accommodate them, and then you give them the best death you can. That's the way I reconcile it. But it's still, I mean, sometimes I feel like I'm telling myself lies, you know, that you know, how about I gave them the best and the death was as beautiful as it can be, but still nagging me that I shortened their lives, and if I wasn't there, or if it was not for my own need as a researcher, then those animals wouldn't have had to die sooner, even though they would die anyways. So they're like a permanent questioning. I never get, I mean, well, you know that very well from our talks, but I never find a good answer to that. So I think I need to keep experimenting and keep trying to rehome animals and find a better and better way of doing research. That's the way I go.
Thanks. I know this is a very difficult question, I think, for all of us. So yeah, really, thank you for sharing. I think, so before we go, I think one last small question from me, because I know that you did some of your studies on commercial farms. So can you actually talk a little bit about how important it is actually to, you know, because you were talking about combining fundamental and applied approaches, and how is it, you know, very important to actually do some of the studies on commercial farms, and what are the impacts you're actually bringing to the farmers maybe?
Yeah, absolutely. So working on commercial farms was fantastic. I really loved it. First of all, because I learned so much from the farmers and from, you know, sticking around, like following them and seeing the way they work. It was great for me because it's a great source of data when you try to think about animal welfare, because you need to see those people in action. You know, what are their everyday life, what do they have time for, you know, because if you see them, if you follow them from 8 o'clock until 5 o'clock, and at no point of time they you know, they sit and do nothing, then how can you ask them to do something more? You know, it seems like their life, you know, their day is already full. So first, yeah, it's a source of inspiration. And second, I love talking to them because I feel like engaging with the public, well, with the stakeholders in general, but the public, let's say the people that are not in science, engaging with them about our projects is crucial. First of all, for us to make ourselves understand our goals, our hopes, our missions in life. I mean, it's really important that we talk more about it, and it makes the public, the recipients of your talk or of your discussion, more aware of what you're doing. And we are doing animal welfare. It's easy to explain to anyone. Anyone can figure it out. Like, can have the images. I mean, I show you a bunch of videos. Every ethologist has so many videos to show what they're doing and what they mean. And I think this is great education. This is how you make people change, is by telling them the stories of your experiments, of your animals, being transparent about what you do, and being transparent about how you feel about what you do. And yeah, because if you are, then they will feel like being as well, and then they will follow. Yeah.
Thanks. So as we approach the hour mark, I want to thank you again, Océane. Thank you for sharing very wonderful research you have been doing all along, and I'm really looking forward to your future results. Hope we will have the opportunity to invite you back to give more talks. And this talk will be is recorded and will be available on our Slack workspace and on our YouTube channel later. And just a reminder that everyone, our application for becoming our speaker is open year-round. So if you have any interesting topic you want to share with us, please follow the link in the chat to apply. And our next planned webinar is on the 5th of June by Holly Vickery on goats. So thanks everyone again for joining us today. Thanks, Océane, for your lovely talk. And I hope to see you again next time.
Yes.
Thanks, everyone. Have a lovely day or the rest of your evening.
Bye, everyone.
Bye.